on in fundamental sciences and national security sectors. Her work has been included research and development on integrated computational environments for bioinformatics, physics, computational chemistry, cybersecurity, and more recently, advanced wireless communications. She's currently a PI for the Millstones and M mutant projects.
Sorry.
No, that's okay. Which applies algorithms and tools for the biological sciences to create new and innovative solutions for related cybersecurity problems, thus merging two of her main interests. So I think that's a really great discussion related to the interdisciplinary nature of some of, as we look at a lot of the AI techniques, we're mimicking different biology type activities. So I think it sounds like a great segue. So Elena, the floor is yours.
Thank you. Nice to see you all. Happy to be here. Thank you very much, Noelle, for the invite. I'm excited to talk to you about some of this work that we've been doing here at the lab for a long time. As you heard in that bio, I've been at the lab. Actually, it'll be 32 years next month. So it's been a while. Came here straight out of school. And one of the things that I really love about this job is that I've had many jobs. I just never really had to move or create a new resume. I get to do lots of different things, learn lots of different things in the span of doing the work that I do here. I'm a software engineer by training. I'm in cybersecurity now. Been branching into things like 5G and advanced wireless communications and also high performance computing. And again, the crossover that we get at the lab is pretty impressive to learn all kinds of different disciplines. But I'm going to tell you today about some cyber work that we've done that I'm pretty excited about, that I think is pretty cool. And I'll go into some detail for those of you who are very interested in cybersecurity. I'll start off with a little bit of background about the lab, the National Lab, and how we work, just in case that's new to some of you. And I did want to start just really quickly with a comment about the Women in Cybersecurity Conference. And I wondered if anyone here actually went to the Women in Cybersecurity Conference that was just this past week, or if you've even heard of the Women in Cybersecurity Conference. Maybe raise your virtual hand if you've heard of it. I see the one on video, yay. So good, oh good, a few others, great, fabulous. It is a wonderful conference for folks in cybersecurity this year. There were 1,700 attendees, over half of them were students. I am very happy to give you any information and background on it if you're interested. I'm very involved with the Women in Cyber Conference. This year, one of our keynote speakers was Jen Easterly, who is the new head of CySER for DHS. She was quite a great speaker. There was a lot of great speakers and interactions, talks, and we did a career fair booth as well. So like I said, it's half students. Most go there on some sort of scholarship. A lot of it's geared towards getting women in cybersecurity out into the workforce. So just a shameful plug for that, since I just got back from there and had a fabulous time. Seeing everyone in person too. I actually got to go to in person this year. So give me a holler if you're interested at all in knowing more about that, and I can get you some information to that, and I can get some stuff out to Noelle too. All right, so I'll start a little bit with making my team's work with just a little bit about the National Lab. So in case you don't know, the Department of Energy has 17 national labs who are charged with tackling the critical scientific challenges across the nation. They're diverse in their mission sets, and obviously all over the country. We are up in here, just south of you all at south of the Hanford site at PNNL. But again, they're all over the country. You may or may not have heard of some of these national labs. We all have similar but different missions. We work together quite a bit, doing different things. We happen to be stewarded by what's called the Office of Science at the Department of Energy, but it's not the only thing that we do. And so let me get here. Just to give you a sense of scale for us, we spent about $1.25 billion last year, over 5,300 staff. Those staff are, well, they're all at home right now, but most of them are here in Richland, somewhere at least. We have a fairly large office, but still less than 1,000 people in Seattle. We have offices in Sequim, if you know that area over on the west side of Washington, a couple other small offices, and then a whole lot of people working across the the nation, and several in DC, of course. We are much going towards hybrid workforce. A lot of people are staying home, whether they're in Richland or not. And it'll change a lot of the dynamics of how we do our work in the future, like it has been in the past. As you see here, we're one of the most diverse national laboratories, and the kind of work that we do, whether it be science or the environment and energy, or what we call national security, which is where all of our work is, excuse me. About half of our business volume is in national security. A large chunk of that has to do with the security of the nuclear arsenal that DOE stewards, and that we help with, but the rest is a lot of cybersecurity and related work for all of the government agencies, including what we call the IC, which is sort of the super secret squirrel agencies that are out there. We especially where I work are mission driven in computing R&D. So we do research, we do engineering, we do operations. Also, sorry, I'll click these out. What we don't show here is sort of the loop from operations back into research. So what happens in the real world drives the kind of research that we do. Academia does a lot of very, very fundamental research. We do a little bit of fundamental research and maybe take it to the next level, do some things like applied research, where we take a lot of that very basic fundamentals and really try and make it into something that can be applied to a problem set, all the way into fully developed applications that are run, given to, run to, sold to others to use on a regular basis. So we really do span the whole gamut. My Teams window goes away, Noel, so if you could pipe up if anyone has questions, I'm more than happy to answer questions along the way, or I can certainly answer questions at the end if you prefer. If you do see someone, Noel, that has a question, if you could just have them pipe in, because my Teams window gets buried. Yeah,
I'll definitely, if there's anything in the chat, I will let you know.
Perfect, thanks, appreciate that. All right, so that's a bit of background on PNL. It's a great place to work. Certainly in this area, but really we're very mission driven about trying to solve the nation's problems. Me in particular in the cybersecurity area, national security area, but we're very mission driven and mission focused in the kind of work that we do. More fundamentally research based than necessary operationals, but we do operations. We use that to help inform our research, and we do it in a lot of different areas. So there's a lot of different things to work from and to bring together. So I'm going to talk to you about a set of algorithms and a tool suite that we developed called Millstones. We actually say Millstones. It actually does stand for something. We're working for the government. You get nothing but acronyms everywhere. So if I spit out an acronym that you haven't heard before, please let me know. I tried to get them out of here. We also code name all of our projects because it's what the government does. So we do that too. So this one is called Millstones. And really it provides some algorithms that has some pretty cool capability, especially when you apply it to cyber, which I will apply for you here in just a little bit. It won an R&D 100 award, which there's the top 100 research and development R&D 100 magazine puts out, and we won one in 2019. We have a couple patents on this as well. So what we really do with the capabilities, we can do things that we call comparing sets of behavior, but behavior is defined pretty precisely. It's an ordered sequence of events where the order matters, frequency doesn't. So it doesn't matter that you maybe tried to log in 10 times. That's a frequency thing. It matters maybe whether you tried to log in, then do this, then do that, then do some other thing. What does that pattern really mean? It can be time, it can be a sequence, it can be any sort of domain specific ordering that we can apply these algorithms to. The events themselves are anything that can be consistently labeled and grouped. So it doesn't have to be what we think of as behaviors and clicking on a link or something like that. It can be other things, and you'll see that when I go into a more specific use case here. Things like opcodes from disassembled binaries, log file entries, patterns of net flow, real-time events, etc. We can do this in and out of cybersecurity. Most of my language will be towards cybersecurity though. And we do it using the nature of protein similarity. So I think in my bio, it mentioned that I did some bioinformatics. I did a lot of software in bioinformatics, and I sort of crossed the boundaries between bio and cyber here. And that's what really made this so powerful. And I'll explain that a bit. The use cases for it. So what we can do with these algorithms that we have is we can characterize behaviors, and remember behavior is ordered sequence of events. And then we can cluster them into what we call families. That's what all the different colored dots you see on that picture are. You can think of them as clusters. Family is a biological term. It just means things are related to each other in some way. And we can do that to reduce data for doing faster comparisons. So if I prime you to think about malware and how we do malware, how we identify malware and how we manage malware, for those of you who work in that space, some of this will, you'll start to be making the map between these things. We can also use this capability to find new and similar things. So things like zero-day malware. We can use these sets of algorithms too, where things are very similar but don't have to be exact. And I'll show you why here in just a second. But you do need some ground truth for identifying things. So I can find a lot of things that are similar to each other, but whether those are good, bad is really two things. One, we have to know if any of them are good or bad. And two, good and bad is kind of in the eye of the beholder. To us, something like Skype and Teams looks bad. It's all peer-to-peer, right? We do it anyway, and it's all fine. So we can identify new signals quickly, and we don't have to be looking for something. So sometimes we used to say it was a way to find a needle in the haystack, but sometimes you just find that needle you don't even know you were looking for. And you can use this sort of set of algorithms to be able to do that. We've done this a lot in binary analysis, especially in the world of malware, but we've also done it on NetFlow data, looking for anomalies in behavior of machines based on their network flows, and also based on their log files. So a number of different ways we can use this set of algorithms and tools in cyber. So now I'll get to a little bit of the fun stuff here if we talk about malware, the use case. So if you guys have been working in the area at all or have been studying some of this, malware is not easy to identify and even harder to classify. There's lots of different people out there that have tried to group together similar pieces of malware. VirusTotal, VirusShare, various parts of the government have done it, and it's not the easiest thing to do because most of them look at different factors to try and find similar things to group things by. And most of the detection methods that we have now are not that hard to fool. So the basics are get an MD5 hash of the code. Very simple to break a hash with very simple techniques, change a little bit of code, change a little bit of data. A lot of it's very reliant on exact matching. There are tools out there that do AI and ML on malware, and a lot of the features are things that are also pretty easy to sort of influence and affect so that you won't be detected. There's dynamic ways where you look at, they will call you the behavior of the malware. You watch the malware do its thing, which is fine if you have time for that. But we, especially at the government level, don't have time for that. We get hit, we get bombarded, the agencies get hit and bombarded with all kinds of malware. And we want to be able to detect it as fast as we can. We need to be able to do that in near real time. One thing that you may not know is that most malware detection tools use some lists. If it's a hash or some other output from an algorithm, they use a list of things to compare to. And last I saw, CERT had over 90 million pieces of malware in their database. And you just don't have the capacity or the time to compare anything to 90 millions of anything. Otherwise, that's all your computer would be doing all day long. So what they do is they cluster in some ways based on things. They do try and create clusters of things to reduce the data. The other thing they'll do is age off malware. So if the malware is really old and hasn't been seen in a very long time, they'll take it off the list. Which is pretty simple to go get an old piece of malware, modify it slightly if you want to do something and let it slip through. And once it's detected, it'll get back on the list. But you might, if you're the bad guy, you might get in and get some things done before that happens. So we really looked at needing a different way to be able to identify zero-day type pieces of malware very quickly. And that's what we tried to do with this system. Really quickly, I'll talk about the biology behind this, just so you know we didn't make it up. Without going too much into biology, though, in the real world, in our bodies and cells in general, we use proteins to encode information through genes, from our genes. And proteins are really kind of they're the workhorse of the cells. They're what really make things happen. And if you're wondering at all whether this is the same kind of protein you get in your eggs, yes, it is. I asked that question a decade ago when I was starting to do this work. Yes, it is. And so it's that fundamental to biology, right? So if you remember any biology classes, you have DNA made up of nucleic acids, A, C, T's, and G's. You have proteins that are made up of amino acids. There are approximately 20 of them. If there are any biology majors out there, don't hold me to the exact 20. I realize that there's a little bit of wiggle room in there, but essentially 20 amino acids that make up proteins. And so they represent them when they try and represent protein in silico, they represent them by a letter. That's why there's a string of letters there. So one protein is represented, or one amino acid, sorry, is represented by the letter A. We picked 20 letters out of the alphabet to represent proteins. But the real key here is what's in blue. When the sequence of a protein is highly related to its function, and this is just natural law. And so if you find a new protein, you can sequence it, get all its amino acids in order. And if you can compare that sequence of the protein to other proteins that you already know exist and what they do, you now know what this new protein will do, essentially. The matches don't have to be exact because there's a lot of things like evolution that have happened. But it is a way to figure out what a protein does without actually having to watch it in action. And if you think about malware, that's very, very analogous to malware. So lots of times if you try to put malware in action, it won't behave like a bad guy. It'll know it's in a virtual machine and not do the bad thing, or it'll be very hard to see what the bad thing might be. So you want to be able to look at it in some way without having to actually watch it and have some sense of what it does. So we use this concept of protein similarity to apply to malware, and I'll show you how in just a second. So what we do is we take a piece of code and we disassemble it and just pull out all the opcodes, function by function, and pull out all the opcodes, ignore the operands. So we don't look at the data at all. We just look at the structure of the code at the binary level. I mean, at the lowest level. And we break things into functional blocks, and then we do what's calling bin the opcodes. So there's, I don't know, 14,000 or so opcodes maybe in Intel. So we can't handle 14,000 letters. So we bin them. All the move-like operations go to the letter A. All the push-like operations go to the letter B, or something like that. We end up with strings of these letters for each function that you can see up here. And that's what represents the function of code. It's just those strings of letters, just like they would for a protein. And then there's a whole set of algorithms that we originally borrowed from biology and then completely rewrote ourselves. They're very akin to machine learning types of algorithms. They're a slightly different kind of statistic called Smith-Waterman, but it's a very similar kind of thing. But we use a bit of what we call a scoring matrix. Anyway, it's just a way of saying, hey, if you try and align two sequences up like we see here, but there is a little bit of a gap or a slight mismatch, so we'll take your score from perfect to something less than perfect. And we'll find the best matchups of all these sequences and then give a score as to how well they match up. So you can kind of think of it, it's got 100%, they're exactly the same. If they're about 99%, there's some slight differences. We had to extend some things here, mismatch some things there. So it's 99% similar. That is very coarse-grained of what we actually do, but it gives you the general sense of how it works. And so we can say two things are similar at some level based on this. Now, we originally did all this using biology code. Nothing of it was ours. It was all biology. We would create our proteins, shove them into the biology code and see what came out. We were actually able to identify clusters and families of code and things that were similar, and even identify things that didn't exist in there but were similar. So it worked for us. Then we rewrote it for a lot of reasons to take the chemistry and biology out of the algorithms and make it a little bit more generic for what we were trying to do. What we do after that is create what we call families, but you can just think of them as clusters, like I said. And there's various ways of doing that, both in biology and outside of biology, and make families of things that are similar. And the reason you do that is because you might have a lot of data. Say you have 1 million pieces of code. It's really hard, like I said, to compare to 1 million pieces of code. It takes a long time no matter what you're doing. But if that 1 million piece of code really only represents about 10 families of similar things, and you could represent each family in some way, now you're only comparing to 10 things, and that can happen very quickly. And so that's what we do. We take all these clusters of things that are very similar based on the algorithm I just showed you, and we make families out of them. And then we represent those families either as some sort of consensus type of signature, like we just find the things that are all the same, or we use like a hidden Markov model or something like that to create a signature for the family. And so now again, if we started with 1 million pieces of code and we cluster that into, let's say, 100 families, now we're only using 100 representations of those families. You give me something new, I very quickly compare it to those 100 things. I say it fits in that family. It could be exactly like something in that family. It may not be anything. Well, it'll be mostly like things in that family. It may not exist in that family specifically, but we know it's a member of that family based on all the similarity. And so now I have some sense of what it does if I know what that family does. So hopefully that's making sense so far. That's kind of the basics. I'm going to show you an example kind of a family, and I apologize for the colors on this. When you pull this out of, I think it was IDA Pro, this is the colors that we ended up with. So the details aren't all that important. The things that are important to know is that these are 2 pieces of code out of 2 different pieces of malware, both do encryption. And you can tell by the bluish here that there's definitely some differences. There's certainly differences in, there's differences in the operands here for sure. There's a lot of the instructions that are the same. There's some difference here between the include and the XOR compared to the sub, right? So a lot of similarities, a lot of differences. To us, they belong in the same family. They both do encryption, and in fact, will both produce the same output, if I remember correctly. They do it slightly different ways, so they look a little different once you disassemble them. So these would be grouped together in a family. We'd say they were very similar, and anything that was similar to these, we would know was encryption and related. And as you saw, right, the registers might have been different, the encryption keys might have been different, but for us, these all come together in the concept of family and representing the behavior that we're perhaps looking for. So with that, if you're thinking about now we have binary code. Binary code can often be influenced by compilers and what compilers might do to them. We also went through did some experiments based on compilers. The one thing that at a national lab you learn a lot is the scientific method. And a lot of the research that we do, we use sort of research and experiments to either prove or disprove if we can. We'll put together a hypothesis, even in cybersecurity, and try and prove and disprove them. So we did that in this case. We ran 2 different experiments, one to see if we can detect similar binaries when different options on a compiler were used. So one of the things that I may not have mentioned before, if you're working in malware, one of the things you do not want is a lot of false positives. False negatives suck too, because then you get hit with the malware, but false positives make the defenders go crazy. And it's a little bit like crying wolf. They'll stop to believe your tool if they're chasing down false positives all the time. So there's always a very delicate balance between trying to catch every possible piece of malware and not making the defenders go crazy with running down too many false positives. So false positive is something that's maybe identified as bad and turns out to be okay. And that's not a good thing. So we were worried that if you just recompiled your binary, we would catch it as malware, even though it's already been identified as not malware or something else, right? We would misidentify it just because it was compiled differently, or perhaps because it was compiled with different compilers or versions of those compilers. So we weren't sure how this algorithm would hold up under those kind of situations. And one interesting thing about what we built here was we started out with an idea of taking an existing metaphor even of protein similarity, applied it to real world code and data, and then just said, hey, it worked. And then you kind of go back and think about, yeah, okay, it makes sense why it works. And then you test the heck out of it to make sure it really works. And then you get where you are. So then every time there's a little bit of a different sort of approach, you have to think through how will that affect what we do based on what you know. But the real true way to go about finding out is to test it. And so these are 2 things that we tested. So really quickly, the answers are, yep, we can to some level detect optimizations. There's about 3 groups that we can be sensitive to. One is the lack of optimization. The next is some weak optimization, and the next group are very aggressive optimizations. So we can't tell the difference between O2 and O3 and OFAST. Those are too similar at the binary code level, at the opcode level, but we do differentiate between that group of optimizations and completely unoptimized. The other one is the optimization for size. We don't catch that because mostly malware is written to be small. Most malware writers will write as efficiently as they can to make their binaries as small as possible. So the optimization for small doesn't do much. Doesn't change things for us much. So we did find out that we were naturally sensitive to at least groups of optimizations. And that, but again, that wasn't by design. That is sort of just the nature of the algorithms at work. Not that we designed it that way. And then we did another, like I said, campaign or experiment on different compilers. We used the tool called Nmap because it looks a lot like malware, even though it's not malware, and we couldn't run malware on our high performance computing where we did all this analysis. So we have to use sort of proxy code for that. As you can see, Clang looks different than everything else. I think the interesting thing for us was some of the break we saw between GCC 4.8 and 4.9. If you can tell by the heat maps here, there is some distinction between those. And the distinction isn't between 4.9 and 5, which is you might have expected from the numbering system, at least, that it was a different. So we did some research to find out what was going on there. And what we found out was they previewed a lot of the changes in the compiler in 4.9 before they made them official into 5. So we actually caught that with this system, the differences in those compiler versions. But this basically says, yeah, we can see the difference in compilers and sometimes between versions if they're different enough, which makes sense for us. So can we label something exactly GCC 4.7? Probably not, but can we cluster things that are likely compiled that way? Yes. And do we see some distinctive definition between something like Clang and GCC? Yes, we do. So why would we do this? Why would that be interesting? Well, click the slide, Elena. There we go. There's a lot of ways that information can be used. So it can be used for attribution, which is a lot of what sponsors are trying to do. They're trying to figure out who wrote the software, who wrote the malware. And malware writers are just like you and me in software. We kind of have our ways of doing things. And they know this about some of the malware developers. And so they might know a malware developer almost always uses Clang and almost never uses GCC. So if this one was developed or was compiled in Clang, it might help them identify who wrote it. That's a simple case, but you get the idea. It also can be useful in dividing up a catalog of malware. Like I said, CERT has over 90 million pieces of malware. There's a lot of attributes you could put on there. How it was compiled is just one of them. It can be helped in reverse engineering. Different compilers being used might help you get through the reverse engineering process. And I won't go into the new variants. We never tested it, and it was kind of an out there idea in theory, and we haven't tested it yet. So if you guys work in the world of malware at all, or maybe even some reverse engineering, you may be about to try and say, hey, I bet there's ways this doesn't work. You're right. There are ways that this doesn't work. There are things it won't detect. So if you've been paying attention and you know that we don't look at the data at all, any of the malware that really attacks the data and not the structure is going to slide right by us. So things like DLL injection, we don't catch. And DLL injection is all done at the data layer, at the intersections. All the structure looks the same. Also, we don't necessarily look at the order functions are called. It's not on the slide, but we don't look at the call graphs. When we do this, we just look at the functions themselves. And so if all of the code is pretty much non-malware code, but it happens to be put together in a way that can be nefarious, we won't catch that. We will just see that all the code looks like pretty good code because we're not checking the actual order of the functions, which is interesting because we look at the order of everything else, but not that. We don't keep track of the order of the functions. We don't look at the call graphs for this process. There are plenty of tools out there that do, and we could easily pair up with those, but it's just not part of our process here. And then other things like packers. So the world of malware uses packers quite a bit. And what that means is that the data segment has an unpacker in it essentially. I'm sorry, the executable segment has an unpacker in it, points into the data segment where the code actually exists. and executes the code there. So we don't look in that segment at all. So we will find the packer, but not much else. That said, it's pretty easy to identify pack code and to unpack them. So we leave it up to the infrastructure outside of us to do that work. And then we can go after the binary itself, create a binary and go after it itself. It's also a very fuzzy, lossy process. So if you remember, I talked about how these things are grouped by similarity and how we bin all the opcodes together. So you cannot take one of our protein sequences and recreate a binary from it. One, we don't even bin every opcode that's out there. We ignore many of them. And we don't look at the data. And like I said, all the moves go to one thing, all the pushes go to one thing. There's other combinations like that. There's no way to reverse engineer. So it does mean you have to do some housekeeping, since you can't reverse engineer these things. You can't tell what they are, but looking at them. So you have to be careful about tracking things. We also don't work with small data. So protein data isn't small. Cyber data is not small either. We actually had to do a lot of work to scale up to cyber data, but it doesn't work on small data. So if you have sequences of 10 or less, there's lots of different statistical methods that can help you do the kind of thing you do. You don't need the big hammer that we have. If you only have 100 pieces of data too, there's a lot of statistics you can use out there, even with regular expressions that would work probably as fast as we would. So we're really looking at data scale, at big scale of data versus small scale of data. Somebody asked me one time if I could use this to do something with social security numbers. I'm like, wait, aren't those like 9 digits long? Like I could brute force that for you more than use some sort of crazy heuristics. But that's a whole nother thing. You can apply this in many domains, and we have. And the process really is figuring out what the mapping might be. So we move to A and push to B. What is that mapping? What does each letter represent? Also, I didn't mention earlier, we were originally constrained to 20 characters, 20 buckets of things. Because we were using the biological algorithms themselves, we've since completely rewritten them and now can have virtually an unlimited number of characters. In fact, my engineers one time decided to send me a data file where they had changed all the mappings to emojis. And so all I saw were just strings and strings and strings of emojis. I forbid the emojis from the processing in the future because that looks like we're not being very serious about what we're doing. So yeah, we use letters now, and any sort of ASCII character that's out there we can use. So you have to do that mapping. You have to figure out what that mapping is, and then you have to test to make sure that that mapping does still hold true in your domain. And then I think I mentioned earlier, it says up there, you have to understand the false positive, false negative, true positive, true negative. Like I said, we can tune this. So when I said, hey, it's 100% match, great, that's easy. And then it's a 99%. There's really a score and a number of other numbers that we look at. Does that mean it's malware or not? I don't know. It could, right? How close do you have to be to the family to consider it to be in that family? That is something that can take training. It can take subject matter expertise, and it can determine how many things do you want to chase down that might be a false positive versus catching all the true positives. So if I didn't say it right, I'll try and make sure I say it right now. You could catch nearly every very, very similar thing, but you're going to catch a lot of things that aren't similar, or you can let some of those things go so that the ones you do get are more likely to be true. And hopefully I said it better that time. But it's a bit of tuning for sure. Let's see. Then we have taken this and sort of turned it inside out. We call it binary dissimilarity. Most people call it binary diversity, but I work with a lot of very staunch statisticians that say diversity is among a population, and dissimilarity is when two things aren't similar, which is really what we're looking at. So we have to say it correctly there. It's the same general process that we have. We disassemble and compare. But what we're really looking for is how similar are two things? So whereas before we were looking at, does this thing belong in this family? Now we're just comparing, say, two things. It might be a handful of things, but two things, and trying to put a score on how similar they actually are. or how unsimilar, dissimilar they actually are. And so we were paid to do this by the DoD, and we put this into operation in some of their work, and really trying to understand if any two pieces of code, just how similar they are or aren't at the level that we can see them. We can do this on the 5 instruction sets you can see there, and I believe we've added a few more since this slide was originally put together. And again, this capability is available. I'll leave it up to your imagination as to who might use this or why, because that's the part I can't say out loud. But I can tell you that we have this in operation as well, where we can do these kinds of comparisons. So that's pretty much the general gist of how we took some biology and bioinformatics and turned it into malware detection. Similarity-based malware detection. We can do it in other things in malware. Here's our team, and I put the team up here, not that they're here to get credit in any sort of way, but just to show you the diversity of what it takes to sometimes make this happen. So I'm a software engineer and doing cyber. Chris is our biology expert and does some HPC. We have software engineers, reverse engineers, statisticians. Of course, then we have people who work in UI and or malware experts. And then interns. Cameron, I had stars on there because he actually came to us as an intern, worked for the summer, used some of the development he did on our mutant tool for his senior project, and started his job about 2 days after he graduated from college over in Walla Walla. We do that a lot. We love our interns, and we look at developing them into staff members quite often, and he was a shining example of how that worked for us, and he's now one of our big reverse engineers. So that's it for the technology. I'll run through a few slides that are a little bit obligatory about partnering with us, since I am talking at a university here, and then answer any questions that you guys might have. We partner and collaborate with academia on a regular basis, industry and government, of course, in all kinds of ways. So that's a little bit of sales pitchy slide I'll skip over. We do a lot of joint research and development. We've worked with Wazoo quite a bit in the past in all kinds of areas. We have wonderful internship programs for both undergrads and grad students, and of course positions, including joint appointments with universities. So I'll do a little bit of pitch for interns. We have a few programs. All of our interns are paid, as far as I know. Every intern I've ever met has been paid. We used to bring them on campus for the summer. Now we have remote internships as well. We do bring them on campus if we need to. We can now with the way things are. And we really do look at internships as a way for us to evaluate you as potential member of our team and culture, and for you to do the same as an intern, to really say, is this the kind of work I want to do? Does the mission really drive me? Is the environment the kind of place that I want to be at? So I think we treat our interns really well. We try to give them the best experience that we can, whether they stay or don't stay. Even if they stay with us or don't stay with us, if they end up in working for the government in that or in the national lab space, we're pretty happy about that as well, even if they go to another national lab. Because we're always just looking for good people who can contribute to our mission and work in our culture. And having just come from the Women in Cyber and talking a lot through recruiting event there, our mission is a little different. We're a government contractor. We are really looking at sort of improving the nation, defending the nation when it comes from a cyber point of view, not looking at making the next fancy like button for Facebook. That's not our purview by any stretch. But what you do get is really innovative technology, a whole lot of infrastructure to help innovate in the technology, whether it be cyber or software engineering or that. We get to work with the universities quite often to do a lot of the collaboration where we might need some very academic and fundamental research to be done at the university, combined with our experience with our sponsors and our infrastructure to be able to make something bigger than what we could do on our own, et cetera. So that's kind of my pitch for the lab itself. Hope you don't mind that a little too much. And with that, I will say thank you. There's my information. If you need it, it's really simple, elena@pnnl.gov. You can email me with any questions you might have after this. And I think I left a few minutes for questions. And I was going to go back And I can send these slides to
Yeah,
Noelle as well.
yeah, that would be great if you can send
Yeah.
those to, if you can send those to me so we can share. Because some of the
Definitely.
students can't be here. You know, some people have conflicts, so
Sure.
it's great for them. Yeah, a couple
Sure.
questions. So the first one is, it seems that you mainly use these algorithms for identifying malware and compiled binaries. Have you tried this for, say, JavaScript malware?
We have not tried it on scripting yet. Scripting is a whole new world that we have not gone into. We would have to tokenize the script in some way. The order would work because basically the script things are in order. I think it's the tokenization problem of the different pieces of script and being in a particular order, and that for the sentences ourselves to be long enough, like to pull everything together, might be a bit tough for us. We haven't been challenged to do that yet, but it's certainly something that we've talked about many times. But very good question. And I threw this slide up here too, because this is a journal article that we wrote, has a lot of the technical background on the program itself. So, but good question about the scripting, and we do get asked that sometimes.
Great. The second question is, can you give us examples of one or two families of malware in terms of their identifying characteristics that
Sure.
you have identified using this technology?
Sure. So at the highest level, you can think of things like Trojans or worms and those kinds of things. But there's definitely families of how they are built and how they're developed. We actually found a piece of malware on P&L systems that were not in the virus total virus shares, and then we sent a cert, and we named it Unicorn Assault because it was a version of the Unicorn Trojans that had not been seen before. So we got to name it, and we named it that. I don't know why. I wasn't part of the naming scheme, but that's just one sort of piece of new zero-day malware that we fit in. It's really based on, is it doing encryption along with other things? One of the things that we want to take a good look at is if we can really put into families ransomware. We could certainly identify the encryption of data through that, but people encrypt data all the time. So, are there other constructs around the encryption of data with the ransomware that we might be able to find as well? And we haven't researched it yet, but we'd like to. Other questions?
Noah, you want to ask your question?
Yes. Well, first of all, I'd like to thank you. This has been very interesting. As a matter of fact, reading the notes, the PDF they sent out to us, I figured that it would have something to do with genetics and the base pairs. But I have a question about your neural network. I'm curious, since and looking at the two, you'd be like, okay, how similar is this piece of malware to this other piece of malware, and would it be beneficial to ensemble the two neural networks of comparing similarity and dissimilarity with two pieces of malware?
Certainly. The use case for that is interesting, and I can think of some use cases in places that I can't say out loud here for comparing the two. So finding some things that are similar, and then how similar are they can have a lot of impact. So we looked at how dissimilar they were, but it's not quite the inverse, but it's a similar operation to figuring out the dissimilarity versus the similarity. And the distance between those could be interesting in some use cases, but I won't say those out loud. But yes, it's a good thought. Some other questions?
Okay, I see one more question in here is, in your experience, what is a bigger priority? Mitigating damage caused to the company or prevention and discovery of new attacks?
So that's going to really depend on the sector you're in and the kind of work that you do. But I would say from the government point of view, certainly detection and prevention is a lot higher priority than the mitigation. They'd much rather defend, and they're better at defending than necessarily mitigating. What I mean by that is there's a lot of people working in that space. So if you're a small business, you may not think so, right? If the mitigation is going to kill you, that might be the higher priority. But I would say in the government space, it's very much detection and prevention of any sort of attack or malware. You have to remember too, in our world, very often attacks are slow and low. So they come in, they sit, they look, They don't make a lot of noise. They don't steal things. They watch and they learn, and they use that against you in other places. And so preventing that, sometimes you don't even know you've been attacked. So the mitigation for that might be in a completely different place, right? It's not necessarily just, you know, they've gotten all your business data or they're ransomwaring you, things like that. The attacks might be much more subtle. So you definitely want to be able to identify those and potentially prevent them. Sometimes you don't want to completely prevent them. So if you think about it, sometimes you want to know they're happening. You want to watch them as much as they're watching you. You might want to guide them into certain places where you can figure out who they are, what they're doing, and why they're doing it. So you might want to know they're there, but not just stop them from being there. That happens a lot, and it's interesting, right? You know, you can see an attack coming and you let it happen. You control it, but you let it happen so you can get more information about the attacker. Yep, that's the kind of fun craziness we get to do sometimes.
So those are the honeypots you have out there to get people.
Yep, honeypots sometimes doesn't have to be a complete honeypot. Sometimes you'll just watch them and then you'll stop them when they get to a point where you want them to stop, but you'll let them get farther than you might think. So that you might gather more information about them, about what they're trying to do, how they're trying to do it, and maybe even why they're trying to do it or who they are. Yep. Other questions?
Great. So we've run to our time. Is there one last question anybody has?
I do have one.
Sure.
Thanks, Elena. It was a very interesting talk. I can see what the biology and expression comes in, and it came in very nicely. My quick question is, I can see where the sequences would come, and you can compare, you know, scores and things like that. I wonder if what you did also played into graphs or networks they're used in biology, and if you very quickly can say if that's part of the things they do.
Yeah, I think so. There's a lot of work in biology on graphing some of these protein sequences, graphing the families and how they're related. I didn't go into that because I only had 50 minutes, but there's far more in there. But certainly, so biology, these protein structures have evolved over time. That's not true. That's one of the parts of it that doesn't hold true in cyber. It's hard to create a whole, what we would call a family tree, because in cyber, you can jump back over there, grab that thing and go somewhere else. So the family tree looks like a hot mess compared to sort of the normal evolution of, say, something like a protein. But we've certainly looked at things in graph forms, especially in and around the clustering for families, if that's what you're talking about. So we did a lot of graph analytics and tried to do clustering for families, community detection, and things like that. And then a lot of graphing around how things are just related to each other in general. Again, it gets a little tough in cyber because I can go to something 20 years ago, grab a piece of code from there, go to something else from 10 years ago, grab a piece of code from there. And it's not kind of the linear progression that you might get in true evolution. So that's one place where a metaphor breaks down.
Okay.
Did that answer your question?
Yeah, those are the kind of things, but there could be newer things you could do too. But you've touched on what I wanted to know. It is in your space, is what it means. And if I have other things, I'll catch up other times. Thanks.
Definitely. I'm happy to answer more questions if people have time, or if you have hard stops, I'm good. But I'm done for the day after this, so feel free to ask any more questions that you have. Everyone else is done for the day, it looks like. It's a long Monday.
Thank you.
Yeah, it is. Yeah, thank you so much for your talk, Elena. We really appreciate the great information that you provided. And yeah, if you can send me your slides, that would be great. We will post those with the presentation. So we appreciate you taking the time today and really providing some great insights. I think